This is production of a truncated, non-functional mutant protein. You know when you start to draw a picture and sort of give up halfway through? Maybe you lost the pencil you were drawing with and had to finish with a crayon? What normally happens is you have half a picture that looks great and the other half not so much. Sometimes when coding for proteins, there are bumps in the road that totally change the outcome of a protein. DNA is transcribed into RNA, which is then translated into proteins. Therefore, a point mutation can completely change the sequence of amino acids that make up a new protein. Mutations can occur in many ways, such as point mutations, frameshift mutations, insertions, deletions, etc. Mutated proteins often will see improper function due to a change in the sequence of mRNA derived from DNA. A truncated or abbreviated non-functional protein, mutant protein, occurs by a point mutation or mutation of a singular base pair in DNA. Given the example you see here, we are going to look at how either an insertion or deletion of a single base pair can mutate and completely alter a protein. The normal and mutant proteins are as follows: MET, TRP, PHE, THR, LYS, GOY versus MET, TRP, Eileen, HIS. To figure out what mRNA sequences code for what codon, we look to the Universal Coding Dictionary. Let's start with the normal protein. We know that MET is methionine, otherwise known as the start codon, which is AUG. Next is tryptophan, which is UGG. Phenylalanine can be either of these two listed. Threonine has 4 options. Lysine is either of the two listed. Finally, glycine also has 4 options. Since our first 3 letters are the start codon, the reading frame of 3 nucleotide bases starts with AUG and ends with GGC. This protein appears to go on as there is no stop codon indicated. For the mutant protein, however, we only see 4 amino acids produced. Where the 2 first codons are the same, we notice a change where isoleucine can be any of these 3, and histidine can be either of these 2. That leads us to believe an insertion or deletion of a base pair in the coding DNA caused a nonsense mutation. This occurs when codons within a reading frame are altered so that what used to be an amino acid is now a stop codon. This causes the protein chain to be shortened and can inhibit the protein from doing its job, rendering it non-functional. The change from phenylalanine to isoleucine is where we notice either an insertion or a deletion of a base pair. Up until then, each sequence can be read as AUG, UGG, starting in the 5' position. When looking at the potential codons for the third amino acid in the normal protein versus the third amino acid in the mutant protein, we notice the normal one has to start with a U and the mutant has to start with an A. This makes it pretty obvious that we're looking at an insertion of an adenine in the mRNA of the mutant protein. The following highlighted letters line up for each reading frame, just 1 letter off. By reading this mutation by the original reading frame, we can see that the mRNA coding sequences for each protein are as written and that the letters line up after the insertion. AUG, UGG, UUC, ACU, AAA, GGG versus the mutant AUG, UGG, AUU, CAC, and UAA. These are my sources, and all the illustrations were done by myself.